So Paul Olivier, and okay, now we have the Gis. Hello all. I hope you're all doing fine. And with Mary, we really prepared our session in a very, very robust manner. So we have rehearsed this like... So the important thing that needs to be said here is, and I will hand over to Marie so that she can introduce. So it's a collaboration between Arapi, GIS Autism, and TND. So I suggest that you say a few words in the context of this call for communications, call for papers. So we are in the second part of the Université d'Automne, and the young researchers that you are going to meet today, one of them is the one who obtained the Young Researcher Prize after the first part of our symposium last year. There were lots of applications from young researchers and a lot of very exceptional applications, because we started from neuroscience with animal models and also human and social scientists, humanities. So the diversity in the proposals and the quality of the projects presented or the papers presented was really made our task difficult. We had to choose from a list of very, very high quality. So, we had rich people's problem, and it was not a consensus choice because we were each defending some very, very high-level presentations from the very fundamental neuroscience to applied sciences in the clinical term. And the level is really growing or getting up. As a young researcher, so the level is increasing, means that people are going to be much more interesting than what we were. So thank you. And now that I'm just relaxed here, I can probably introduce our first participant. So now Cindy Bokobza. Cindy, you are a postdoc working at the Pierre Debré Cindy Bokobza. So you received the prize. You were awarded the prize last year. So first episode of the Université d'Automne. And let me hail, say hello to Baptiste and Lauriane. So we really had a great program 1 year ago. So Cindy, your presentation was really, really interesting to the jury last year, because these are studies that are quite new, because you're going to bridge the gap between with an approach that is very clinical, fundamental on animal models, the bridge between what is happening in the very early stages of neurodevelopment with what will take place in the old age. And this is what was spoken of this morning. Cindy, the floor is yours. Thank you. And there is a lot of pressure from all the praise you have just given. I'm very honored to be standing here in front of you. It was an honor to be selected last year, and a great honor to be able to present my work. And as Paul said, I come from a lab which initially was focused on prematurity. Prematurity, what is it? Prematurity is any birth that happens before 37 weeks of pregnancy, any birth that takes place before the beginning of the 9th month of pregnancy. That represents 15 million of births worldwide, 1 out of 10. And it is one of the primary causes of death in newborns and infants before the age of 1 year. Several causes. We have a premature rupture of the membranes for 25%, 30% of medical triggering. In the case the life of the fetus or the mother is in danger, either pharmacological or C-section induced. In 45% of cases, these premature births will come just without any aggravating factor or visibly aggravating factor between the moment the mother is in the hospital and when it was started. But there were risk factors. A previous pregnancy that ended in prematurity, if the mother has been the victim of domestic violence or especially strong stress. But in most cases, we realized a posteriori that there were maternal-fetal infections. So there is a real context, in a context of inflammation that leads to the premature delivery. Actually, part of these children will develop encephalopathy of the premature child, brain lesions. All the lesions in the white matter and on the gray matter that is associated with the prematurity. On the white matter, the lesions are the ones that are the most described, where you can see that on the MRI, you can see the major lesions on the white matter, and that will lead to heterogeneity of the late deficits, cognitive or learning deficits. And then we realize later on that there is also a lesion of the gray matter, which are secondary to the lesions to the white matter. And we have been able to demonstrate with MRI images that these lesions on the gray matter could induce defect of brain connectivity. So where do we stand? We are with the Arapi. We are with the Arapi here. So you know the criteria of the DSM-5 with social interaction and communication and repetitive and restricted behavior. And what's the link with prematurity and ASD? Actually, we realized that ASD leads to a major risk of appearance of or development of ASD signs at the age of 3. And this risk factor has been reevaluated to increase if prematurity is related to inflammation. So the prevalence of prematurity is ASD is really high. But the old premature children born premature who will show some ASD do not present these criteria. We don't necessarily have the restricted and repetitive behaviors, but the two other criteria. Why? So this is a brain, nice cookie. It's a picture that I took from an old class book, and I love it. If our brain was a cookie, the chocolates would be neurons, and the glial cells would be the pasta, the dough. And what are the glial? Everything that is not neurons are glial cells. First, we have the oligodendrocytes, cells that are there to accelerate the transmission through the neurons of the electric signal. And then we have the astrocytes that are the feeding cells that bring the nutrients to the brain, and the microglia is the protective cell that really makes sure that everything happens fine. So a post-mortem of a premature child. So we have lesions and we have holes in the the first victims are the oligodendrocytes, which do not differentiate normally and will not produce the myelin, and you have holes, literally holes in the brain. Interestingly, around these lesions, in the periphery of the lesions, we were able to observe the presence of two other types of glial cells, our astrocytes here in brown, and the microglial cells here in. So our question in my lab was, are the glial cells, and especially the microglial pro-inflammatory, is going to be responsible for the onset of the lesion? So let's go deeper into the microglia. What is it? It's my favorite. It's the soldier in the brain. Why is it the brain? Because it's the only single immune cell that is present in the brain. Why is that interesting in the context of prematurity? It's because it's going to proliferate and differentiate and play a major role in neurodevelopment during the third trimester. That is the moment when prematurity will occur. The microglia has a major role in the normal development of the brain because it's going to promote the formation of the myelin. It's going to be responsible for the what Boris said this morning, and it's going to participate in the creation of the neural networks. But as I said, it's an immune cell, and in case of an insult, it's going to become toxic. In case of neuroinflammation, neuroinflammation is inflammation of the cells of the brain. It's going to induce cells that are toxic for the other brain cells, and it's going to block the maturation of the dendrocytes. And this is why you don't have the normal quantity of myelin in premature babies. The interesting thing is that in postmortem studies of autistic or premature, we have pro-inflammatory processes taking place in regions that are really, really neighboring. So it seems that this plays a major role in the lesions. And at Robert Debré, for 13 years now, there is a murine model. I don't really work with patients, so I work with mice. And we inject in the first 5 days of life a interleukin 1 beta. Why this window of the 5 first days of life? Because in terms of brain development, the equivalent of the third trimester of pregnancy is this first 5 days. This is when you need to look at the brain for prematurity signs. When you look at it, the first thing that you look at is, are we able to find these lesions on the white matter? Yes. So DP5, the phase at the end of the inflammatory phase where you have lesions of the white matter, and P15 when myelin production is well engaged, we will see that it produces much less myelin due to the blocking of the oligodendrocyte maturation. So what they did before I got in the lab, they injected the animals with gadolinium. Gadolinium is a small molecule that is used in neuroimaging. And actually what happens is that this is going to actually suppress or eliminate the pro-inflammatory microglia. And we observed the animals, and what we saw is that normally when you have a microglium, which is going to become pro-inflammatory, we have the myelin production processes that are blocked. But when you remove this pro-inflammatory microglia, the myelin production happens properly. So this means that it does play a real central role in the lesion. And I had two questions. My first question was, does the neuroinflammation promote the onset of ASD signs in the young premature baby? For us, it's a mouse. Okay, it's TSA-like. It's not TSA. It's not ASD. It's ASD-like in my mice model. And the second question is, does the neuroinflammation promote the appearance or the onset of pathologies that are... How can we assess this autistic or ASD-like behaviors in our mice? We evaluate, assess communication. You have to know that the, murine models, they speak, the mice would communicate, and they emit ultrasound calls. That is, they vocalize so that they can communicate with their mother and their siblings, their baby brother mice and baby sister mice. So this image is representative of these calls. And as you can see, we have specific patterns, and we have tools in collaboration with the Institut Pasteur and Thomas Bourgeron's team. We have been able to use these tools to identify these patterns of how the animals vocalize their calls and see if they can vocalize the same way. The ones that are exposed to IL beta will vocalize less, and the few vocal calls that they make are shorter, which means that they have a lesser communication. They communicate in a different way and in a not a way that is not as good. And we also have behavioral tests that will enable us to assess the social interaction in the adult subject or the young adult. So here with young adult, about adolescents, mimicking adolescence in humans. So here we have this square arena, and there is a small transparent box in a corner where we have either a foreign mouse or an inanimate object. And in the middle, you have our test mouse. And what we want to see is if the interest is directed toward the foreign mouse rather than the inanimate object. And this is what you see. My foreign mouse, my test mouse is going to spend much more time with the foreign mouse than with the object. But with animals that are exposed to the IL beta, the test mouse spends much less time with the foreign mouse, but and even spends a lot more time next to the inanimate object. So the interesting thing was that the animals exposed to the IL beta will go in one corner of the arena and avoided looking at the foreign. So there is the gaze avoidance in the animals exposed to IL and beta, IL-1 beta. But the interesting thing for us was that, like with children that are born premature, they do not have repetitive behaviors. And then we assessed brain connectivity after exposure to IL-1 beta. So this is kind of state-of-the-art technique, imaging with echo Doppler, ultra-fast echo Doppler. Echo Doppler is like the sonograms that you do during pregnancy. It's the same thing, but much more sensitive. So thanks to ultrasounds, we are able to assess the movement of the blood inside the brain. And we look at two regions. Here we look at the hippocampus in blue, and we compare with the hippocampus on the other side. And we can see that the signals from the blood are, during the acquisition time, is somewhat synchronized, meaning that the two regions work synchronously. But if you look at the hypothalamus, you see that the signal is not exactly spot on superimposed. So it means that hippocampus works less well with the hypothalamus. Thanks to this, we can assess what we call connectivity matrices, and the region that communicates best with it. And we have a gradient, the hippocampus, left hippocampus correlates a maximum of the right hippocampus. But when we were doing this connectivity matrix, the signals were not exactly the same. So what we did was evaluate the effect of the inflammation. We compared these matrices. If it's blue, it's hypo-connected, lower connection, much weaker connectivity. So here we have a problem of brain connectivity. And then when I arrived in the lab, what we knew very well were lesions of the white matter in our model, but not much knowledge about the gray matter. So my whole work was on getting to understand and know the gray matter better. So what we could see was that there was an electrical activity which was significantly higher in the animals exposed to IL-1 beta rather than the controls. And the reason for that, it's due to the fact that the IL-1 beta animals, here we have assessed the density of synapses on the surface of the neurons, and what you could see in IL-1 beta animals is that they had more synapses than in the control subjects. Why? The question is really, why do we have this issue? And this is when we go back to my pet mice, my pet model of mice, which I really love. So this microglia, which is microglium, which is an immune cell. So what you see with the two red clusters, 1 and 3, these genes are just pro-inflammatory cytokines. And all the toxic cells that can be released. But when the microglia will release all of these dangerous genes, it will sub-express or tone down the other. When you look at the synaptic pruning, which is a major neurodevelopmental function of the microglium, so the synaptic pruning requires it's supposed to do it at week 2, but with the animals under IL-1 beta, they're not pruned by the microglia, so they stay. From these concepts, so we were able to demonstrate that we had lesions at the stage of adolescence. But when I arrived, my lab, nobody went further than the 2 months. But because after 2 months, they were no longer histologically observable data. But for me, there were no more lesions after 2 months, so nothing could be observed. But for me, I was really interested in what was done in the microglia in neurodegenerative diseases. So if my microglia is already damaged, will this have a consequence later in life? And for this, I used concepts that were evoked this morning, the concept of inflammaging, this notion that there is a chronic inflammation that is silent, that will accumulate in the course of life, and also the notion of cellular priming which is defined by the fact that the immune cells will respond faster and stronger if they're exposed as a second stimulus, as a secondary stimulus. So we have this major inflammatory event, which is prematurity at the beginning of life, and this chronic silent inflammation throughout life. And that's the combination of these two factors represent a risk factor to diseases or conditions related to aging, diabetes, obesity, and cardiovascular. So we looked, we observed 7-month-old mice, which corresponds to a certain age or 40 and onward in humans. So the animal will spend as much time with the foreign animal, but the decision to go toward the object or toward the foreign mouse is a much more difficult decision to make. So we coupled this a double field, just the arena without nothing in there. So we observed the animal and its locomotion. At 7 months, we have a locomotor activity that is much more important and that can be related to hyperactivity phenomena. And the behavior disorders that we observe in our murine models are not necessarily what we see in adolescents. Now back to our imaging, and we did the same thing at 7 months. So what we see is that we have regions that are still hyper-connected, the hippocampus. So a major region in our model. And here you have these two red squares representing the hypothalamus. So at 7 months, hyperconnectivity, which is really interesting. And you will see why it's very relevant. For the white matter, we will have analysis in the only region where we see a lesion that persists 7 months after the inflammation is not at all the callus, corpus callosum, but the cingulate. I'm sorry about the quality of the photographs on the screen. What we saw is that we have the neural cells. We see that at the level of the hippocampus, we have a significant increase of the number of neurons and much less in the hypothalamus. So you have lesions of the gray matter, which will be specific at 7 months. And this is something that really did not expect. At 7 months, it was very region-specific. And in view of the white matter, we only had an impairment or an alteration of the cingulum with a hypomyelin production, and here also with a Western blot. And then we have a reappearance of the lesion of the white matter, and at 7 months we have this hypomyelination, and this actually corresponds to the death of the oligodendrocytes at 7 months in the animals. So the base concept on which is that we have inflammaging, and we need to be able to demonstrate that this slow, silent, chronic inflammation is present. So we looked in the blood if we had an activation. Yes, indeed, we have the myeloid cells. The immune myeloid cells in the blood are particularly active, and they will release RUSC's pro-inflammatory cells only with the IL-1 beta animals. But now, what about the microglia? It is also inflamed, and we have the traces of inflammaging in the brain. And here it's still an analysis of the transcriptome, that is the expression of the genes. And at 7 months, we had about 100 genes that were dysregulated. And we then had an increase of... So it's no longer developmental functions, but physiological functions of the microglia. But this transcriptomic signature for the expression of genes was not translated into a functionality. Actually, we had the same number of microglia in the hippocampus and cortex, but big surprise, we had much more microglia in the hypothalamus. So at 7 months, we have a signature pro-inflammatory in the expression of genes, which does not necessarily translate into some functional alteration. So this is on the brain. And the first sign that we saw in the behavioral test is a very simple physiological parameter. The animals are really fat, much fatter. And back then, we contacted people who had worked on the French cohort EpiPage 2, because this cohort already highlighted the relationship between prematurity and ASD. And we asked them if they could look if the premature children will be overweight. They looked at the age of 5, and it was possible to see that there was a risk of developing obesity or overweight at 5 in the premature children. And if the prematurity was related to a pre-inflammatory syndrome, it was correlated. So we would monitor the weight progression, and we see that the red line, that is, the animals become overweight, and it becomes major at the age of 7. And this excess weight is associated, and only for males, lack of tolerance is related to lack of tolerance to insulin. Normally, blood glycemia should get lower, but with those who were premature caused by an inflammation and analysis of the tolerance to glucose, we were expecting a peak, and we find it again in the IL-1 animals, higher. The peak of glucose was associated, but only in the males, with hyperinsulinemia. If you associate overweight with intolerance to insulin and hyperinsulinemia, they have type 2 diabetes. The question is, can we treat the animals with IL-1 beta? These formerly premature individuals, can they be treated with metformin, which is the gold standard treatment for type 2 diabetes? Can we treat our animals at 7 months? And we saw that we were able to correct the after treatment, the hyperinsulinemia, in the animals exposed to IL-1 beta. And by conclusion, I hope that I was able to demonstrate that prematurity is a really crucial challenge, and it's a risk factor for developing ASD, and mediated with the inflammation, the perinatal inflammation, that will promote the apparition of ASD-like behavior in the young individuals. And this is related to the fact that the microglia does not do the synapse pruning, which is one of its major protecting functions. And the interesting thing is that there are consequences, long-term consequences of that perinatal inflammation mediated by the inflammaging. And these formerly premature individuals will present new types of cognitive deficits that we need to try to understand. And the reason for that is that the microglia, which is still activated, is dangerous for the brain, which already has suffered lesions. This entails risks of neurodegenerative diseases and something else which didn't expect, which is metabolic disorders. So what I wanted to highlight with this project is that we know that children with neurodevelopmental issues and associated with autism, they will have a problem at school. Stigma because of their overweight. The life of these children can become really, really complicated with the confluence of these problems. So we need to work on food intake, and this is something that we can talk about with the families and the clinicians so that we can avoid down the road that they will be stigmatized. And of course, I will thank the people of my lab, my students, of course, Pierre Gressant, whom you will meet soon, and my students. And thank you for your attention. Thank you, Cindy. So you have you have your pet cell, your favorite cell in the brain. I wasn't here this morning, but we were speaking about genetics, and now you show that in the mechanisms that are at stake, the microglia is really, really complicated, and this is a change of paradigm that has occurred. The shift has taken place in the past 15 years. We realize that the conductor is the microglia, not the... So, and you quoted Geraldine Favre, and really, I will say hello to all the neuropediatricians. And you're showing that there is an impact, and we focus a lot on the early stages of neurodevelopment. But I was not here this morning, but I think that Stéphanie Mion was also giving a very powerful presentation. And now you say that we need to look beyond and what is happening. So Courchaine 4 years ago was speaking about the systemic challenges, looking at what was taking place elsewhere in the organisms. The thing is that I'm not going to talk about neurodevelopmental disorders. We focused for a long time on disorders and cognitive problems, and everything else was less important because, you know, the child was premature. So if the lungs were not really, we know about this, and it's not that terrible. So my child was premature, and he was really small, so I want to give him a lot of food, everything that he wants, chocolate, candies. But short term, if you focus only on the small child and puberty, you don't necessarily see beyond. The child can become happy, but beyond that stage of early puberty, when the person grows old, this will have an impact. So I had attended a symposium of clinicians on aging in the American aging cohorts. There was a realization that the children with prematurity, once adult, are at risk of developing type 2. So, René, my dear friend, you have a question. Metformin. Did you try the new anti-diabetics, the semaglutide? And no, we haven't for the moment. For very simple, metformin is an agonist of the CPAR gamma, which is an anti-inflammatory receptor. Metformin was a good target because on top of reducing diabetes, it also reduces the inflammation. But actually, it does not work in our model on the inflammation. It works only on the... so we really need to look at the inflammaging side of the question. Hello, I'm a neuropediatrician in Guadeloupe, and I would like to know if the mechanism is similar for delays, in utero delays, growth delays, because in Guadeloupe we have a lot of inflammatory diseases and we have a lot of SCP4. Maybe that's multi-factor, but probably also related to the chlorhydricon. I would like to know if science is also looking into the growth delays. In our lab, it's the same team, but another group. It's the team of Olivier Beau. What they did was induce an intrauterine, in a murine rat model that were fed with hypoproteic food. And we had animals that were really smaller at birth. And we would look at the lesions of the white matter. So we had to couple the hypoproteic diet with small, minor injections, small injections, much less than in the mice model, but once or twice a month, the IL-1 beta, and we could find the inflammation. The mechanisms on the white matter are the same. So in view of prematurity, we see that microglia really plays a major central role in all the prematurity models, the hypoxic. So it plays a major role, the pro-inflammatory microglia. She was not speaking in the microphone. In the beginning, I was talking about the maternal fetal infections. When we are in an inflammation condition, there is a priming of the cells to be more active if we are going to have a real inflammatory syndrome. So this really plays in case of prematurity, obesity of the mother is a risk factor for the premature delivery. Hello, I am a mediator in the Landes, in the southwest of France, less concerned with from a distance by ASD, and you were talking about the relations between ASD and type 2 diabetes. What about type 1? Type 1 is an autoimmune disease, so basically that implies the immune system. But the interesting thing in human cohorts, American cohorts, they saw and they could associate with type 1 and type 2. But in our model, we just focused on the fact that we had intolerance to insulin or to glucose with hyperinsulinemia. I have, sampled pancreas to see what is taking place in those key organs that are implied in the development of diabetes. But we know that in man, at least, we have an association with a risk of developing diabetes type 1. So we are also talking about the pro-inflammatory context, so that a child who is not premature, but if we detect an ASD, can we say that the origin was maybe in a pro-inflammatory context? No, honestly, that will depend on each person and the clinical picture and the context. Well, we had this conversation with Boris during the coffee break. Historically, autism is a genetic disease, and any genetic test will enable seeing how these factors, environmental factors, will interact with the landscape, the genetic landscape, which can be more or less altered. But my hypothesis, and it's just me and my little bit of genetics, but I'm not an expert, far from there. The idea is that in reality, we can have a small mutation that does not necessarily have a basis consequence. But coupled with an environmental syndrome, it's going to be overexpressed and will be all the more dangerous. So this is a matter of patient-by-patient case. So patients with an ASD who were born not at all premature, but at the end of the term, maybe they didn't have some, but maybe hypercaloric diet is a pro-inflammatory context. Are we done? Maybe another question? So thank you. Thank you, Cindy.